In today's lab, we're going to have students practice forming a hypothesis. We have 6 different liquids, and students will figure out what the density of each of these liquids is. What they're going to be predicting is which liquid has the lowest density and which one has the highest density. And in order to help students with this, we have given the molecular formula of each of these liquids for them to analyze. Okay, so each group is going to look at these 6 liquids and they are going to state which has the lowest and then higher and higher until you get the highest density. The statement below is the students are going to write down their hypothesis. They're going to state why they think the densities they predicted will be in that sequential order. Each lab group will receive one of the liquids to analyze. This lab group was going to analyze ethanol. They have a big jug of ethanol here. They're going to be pouring a small volume of this ethanol into a beaker from which they are going to run their experiment. So I'm going to be putting about 10 mils of ethanol into this beaker and don't need to touch the stock bottle anymore. Okay, so every group is going to receive 8 Eppendorf tubes. And one of the issues with the lab is that we don't have enough balances for each group to have their own. So groups are going to have to share balances, and therefore we're going to have students label Eppendorf tubes. We're just going to label them A, B, C, D, E, etc., so that they know which tube is which in case one of these Eppendorf tubes has a different mass than another. So I'm just going to label these first 3. We have the balance turned on, set at 0. Just going to put the tube on top, and I am going to record the mass of the empty tube on the data sheet. Okay, it looks like it's 0.98 grams for that one. And again, I'm not positive if all of these are going to have the same mass or not, so definitely want to weigh each tube individually. Do one more for this demonstration. Tube C also has a mass of 0.99 grams. Okay, we'll put the tubes back into the tube rack, and now we're ready to measure some volumes. The first volume is going to be 950 microliters. And this pipetter is already set for 950 microliters. This is a P1000 pipetter, so it measures between 100 microliters and 1 milliliter. Okay, so we're going to grab a pipette tip. We just basically push down against the box. Pipette tip slides right on. We're going to now press the plunger down to the measuring position. Then going to dip the pipette tip barely underneath the surface of the liquid, and we're going to slowly measure the liquid, and then we will allow the liquid to run down the Eppendorf tube, and then we'll close it up, and I am going to change the volume to 800 microliters, which is the next volume that we need to measure. And we'll do the same thing. We can use the same pipette tip because we are measuring the same liquid multiple times. So I'm just going to press the plunger down to the measuring position again, dip the pipette tip into the liquid, slowly release the plunger to measure the liquid, and then we're going to dispense the liquid into the Eppendorf tube. Okay, so I will measure one more, which is at 650 microliters. I'm going to dial it down to 0.65, which is 650 microliters, and measure this in the same way that we've done the others. And again, I'm making sure that I'm putting the pipette tip against the side of the tube so that all the liquid drains out of the pipette tip. Okay. Okay, now we're ready to measure the mass of the filled tube. Again, we'll put it onto the balance, and looks like 1.74 grams. First tube. Second tube is at 1.64 grams. And then the third tube. Measure this one, 1.53 grams. Okay, students will have more tubes than just 3. They'll complete the first 2 columns. And now we can do a subtraction. 1.74 minus 0.98 is 0.76 grams. 1.64 minus 0.99 is 0.65. And 1.53 minus 0.99 is 0.54 grams. Again, students would do this for all 8 of their tubes. Students will now graph their results. They're going to need to label the y-axis and the x-axis. Remember that the x-axis is going to have the independent variable, which in this case is the volume measured. So students should label the x-axis, and then the y-axis is the dependent variable, and that's going to be mass of liquid, which is going to be in grams. Okay, so you're right, should not be grams, this should be in, microliters. Okay. All right, the reason why we're graphing the results is to try to be as accurate as we can on measuring the density. We could theoretically determine the density of the liquid simply by dividing the mass of the liquid by the volume that we measured. And what we want to do is we want to display the graph in what we usually do, grams per milliliter, but right now we have microliters. And so I guess,
grams per microliter.
Grams per microliter. We can just do grams per microliter.
Yeah.
Okay, so what we're going to need to do is we're going to need to label the x-axis with the different volumes that we have. And so we kind of have to count how many lines we have across. 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12, 13, 14, 15, 16, 17, 18, 19. We have 20. And so that means that each one of these lines would be 50 microliters. So this is going to be 100 microliters, 200. So we label our graph here. And as far as the mass is concerned, looks like it's going to be the highest mass we have is about 0.76 grams. So let's go ahead and maybe make each one let's go 0.1, 0.2, 0.3, 0.4, 0.5, 0.6, 0.7. Okay, so we're just going to plot a point, 950 microliters, which is this line, 0.76. Didn't give myself quite enough room. And then we have 800 microliters, which is right here with 0.65. And that's going to be right here. And then we will have at 650 microliters right here, 0.54. Going to be about right here. Okay, so we're going to need to have a ruler, and if we weighed 0 microliters, we would expect it to weigh 0 grams. So we're going to kind of think of that as one of our extra points. We're just going to try to get the best fit line that we can, and I'm just kind of play with it just a little bit. It doesn't quite go through that origin down there. Maybe I should have gone down a little bit further. But anyway, this is going to give us a chance to measure the slope of this line. So what I'm going to do is I'm just going to basically find a couple of locations where the line crosses one of these points. Let's go way down here, and this is going to be point 2. This is going to be point 1, and the slope of a line is y2 minus y1 divided by x2 minus x1, which is going to equal, the y2 is 0.6 grams, and the y1 is going to be at 0.2 grams, and the x2 is here at 750, and I guess I should put down microliters, and for x1 it's going to be 200 microliters, and that's going to give me 0.4 grams divided by 550 microliters. Now we can get our calculator out and figure out what the density of the liquid is.